Good morning, everyone. Thanks. I would like to thank DOP and IMAR for this organization, this event, which I think is very important. I would like to say that I'm a postdoc at IMAR, so an in-house scientist, but I'm based now in Madeira, Ireland. But today I'll talk about two questions I explored during my former postdoc appointment at the Smithsonian Institution, where I worked with Greg Ruiz. I spent there almost 3 years, and I basically worked with fouling organisms. And the two questions I would like to talk to you today is the effects of pollution, metal pollution in invasive species, and also climate change. So I didn't do this work alone, but I was leading this. So the title of the talk is Bioinvasion's Response to Climate Change and Pollution. I'll start with climate change, and then I'll shift to metal pollution. I'll start with some theoretical background on climate change. I'm sure you are all familiar with this, this question. So this is a very interesting paper published in 2010 in Science by Bouchard and co-authors, and it's basically the indicators on biodiversity loss in the last 40 years, starting in 1970 to 2010. So that's what you see on the x-axis. You see the year, 1970, starting in 1970 to 2010, and on the y-axis you see the biodiversity index. So these first three graphs are refer to the state of biodiversity. So the first graph just shows examples with birds, wild and water birds, and you see biodiversity decreasing over time. Same with the red list. This second set of graphs refer to the pressure on biodiversity loss, and you see the ecological footprint increasing over time, nitrogen deposition as well, and of course, alien species increasing over time since 1970 to 2010. This third and last set of graphs in green refer to the response of our response to this loss. So some protected bird areas were implemented, some new policies, national and international, and some aid as well. What is important here is, of course, alien species increasing over time. And this, of course, this phenomenon is taking place every day at a global scale. During my time at the Smithsonian, there was, I was studying that several marine invertebrates from the Caribbean region were migrating north, especially to Florida, South Carolina and Georgia, and this, due to global warming and this poleward range expansion has been coined by Jim Carlton by Caribbean creep. And here are some examples of these Caribbean creep species. The barnacle Megabalanus cocopoma, also Perniveridis, the green mussel, the marsh snail Credonia succinia, and the porcelain green crab, Petrolites armatus. So all these organisms are within this category of Caribbean creep. Okay, so several models on climate change, and this is not new to you, have predicted that in the next 100 years biodiversity will decrease due to climate change, but all these models are based on the temperature increase, and they seem to ignore periodic climate fluctuations such as heat and cold waves. So this is what happened in 2010 during my stay at CERC. There was a very pronounced cold spell in January 2010, and there are several reports of dying animals in this region, manatees and crocodiles, sea turtles, iguanas, and corals. They were dying in this region. So just to give you an example, we tried to find the green mussel, Perniveridis in Florida, especially in Tampa Bay, and we couldn't find none. When we knew that in the previous year there was 11,000 mussels per square meter, and in the following year after the cold spell we found none. So again, from 11,000 to 0, it's a lot, it's very pronounced. Same with corals in the Florida Keys. this is a recent paper, and you see on the first column the corals before the cold spell and after the cold spell. Same with this one of the Caribbean creep species, the Petrolites armatus. I put this graph together. This is mean winter temperature since the 60s till 2010. And we realized that this crab experienced the most severe winter in the last 30 years and the third most severe winter in the last 60 years or 50 years. And here are the abundances, 700 animals before the cold spell to 10 to 12 after the cold spell. This graph shows the water temperature in Georgia only in winter in the last 5 years, from 2005 to 2010, and you see the big dip there in January. So that shows that these crabs experience a very severe winter and unusual. So with this in mind, I had a very simple question, and are extreme cold temperatures, such as the one that happened in 2010, killing Petrolites armatus and perhaps other Caribbean creep species? So let's give a little bit more information on this crab. This crab is native to the Gulf of Mexico. It was first collected in Miami, in Florida, in the 30s. It was reported abundant in the Indian River Lagoon in the 70s, and it jumped to Georgia and also South Carolina later in the 90s. So I performed an experiment at CERC, very, very simple. So I just wanted to mimic this severe winter conditions in the lab. And I had 3 incubators. One was my control. I just put the crabs in the incubator for at 15 degrees for 3 weeks. The second incubator, I reproduced exactly the winter conditions of that year. And I wanted to kill more crabs, so I had an extreme treatment even lower. So I went to 15 to 4 degrees. And this was based in the data collected on the graph above. So at each incubator, I had 3 boxes containing 6 cells. So I had 18 crabs per treatment. I had temperature loggers. The crabs were fed every other day, and water was changed every day. And my response variable was just mortality. This graph shows the data extracted from the data loggers in the incubators. And the green line is actually control, blue is cold treatment, and red is our extreme treatment. You see time and water temperature on the y-axis, and you see that actually the temperature is actually perfectly mimicking what happened actually that winter. So this just shows that the treatments were working, were effective. And this is the simple results. This is a Kaplan-Meier curve, a survivor curve, where you have on the y-axis the survival and on the x-axis you have time. And the green line is actually our control, so mortality was below 20%, so only a couple of crabs died. Our blue line is actually our winter, the reproduction of this winter, and we got a mortality over 60%, and the extreme treatment, all crabs died after 19 days. So main conclusions. This crab cannot tolerate prolonged and extreme cold temperatures, particularly at 4 degrees. Periodic cold events, winters, may be a critical reset mechanism, so reducing the local abundance of these crabs and limit the range expansion of other Caribbean creep species. And I would like to finish with this, this part of my talk, talking about the future and implications for the future. This is a paper by Henrique Pereira. He's a scientist in the Faculty of Sciences in Lisbon, and he published this in Science in 2010. And he basically projected the rate of range shifts in marine organisms for the next 50 years, starting in 2005 to 2050. And the different colors represent the different rate of shift. So if you look into Florida, for instance, you see a green code, which means that he predicts that species, marine organisms, will migrate north at a rate of 1 to 2 kilometers per year. So with this study, we believe that the cold may slow down these non-indigenous species migrations. So temperature aberrations are an important and overlooked part of global climate change. In other words, and that my take-home message to you is, yes, climate change is happening, but the cold is also part of this change, okay? Okay, I'll move to my second topic. Just drink some water. Okay, so my second question was, of course, metal pollution and metals. Pollution can be considered a category of disturbance, anthropogenic disturbance. And there are several studies that say and argue that pollution may promote invasions. Why? Because it increments the availability of resources and also decreases competition with native species. An example from the terrestrial realm with plants. There are some studies that say that there are more non-indigenous species in roadsides when compared to different areas, more plants. In the marine realm, in bays, there are more metal pollution. Why? Because there are more anti-fouling paints and also industrial waste. As you know, anti-fouling paints coatings are applied to ship hulls and also to other artificial substrates. And most of these anti-fouling coatings are very rich in metals, including copper, and mainly copper. Copper is a very toxic heavy metal, and it's very bad for marine invertebrates. It affects its reproduction, growth, and also abundance. I want to show you this. This is an experiment by Jeff Crooks and also Greg Ruiz that they did in California. Also very simple. They deployed PVC plates in San Francisco Bay for a couple of months. After that, they collected the plates and put it in buckets with different loads of copper sulfate, for 72 hours. So they had a gradient of copper pollution, and after that, they put it in the water again, and after a while, they measure biodiversity. This is what they found. On the x-axis, you have the disturbance, so the copper concentrations, and on the y-axis, you have the percent of total number of species. And basically, native species, of course, they decreased with the disturbance gradient, and non-natives, they really didn't care about copper. So you see, they're just a flat line, non-significant, which may indicate that invasive species may be tolerant to pollution and to copper in particular. With this in mind, I had the following questions. Is total diversity sensitive to copper pollution? Is non-indigenous diversity more tolerant to copper pollution than native diversity? And does this change across regions? So I performed the following experiment. We had 2 study sites, one in Virginia, close to Cirque, and one in Panama. As Greg mentioned, Smithsonian has a station there, a very scenic station close to the canal. So I worked there as well. And I created this gradient of disturbance. So I used fiberglass plates, that's what you see there, close to my feet. And I used anti-fouling paint, rich in copper, 76%, and I created this gradient with different loads of paint. So I had 2 sites. I had 4 copper treatments. I had 6 replicates per treatment. I had a block design to avoid heterogeneity, and I exposed these plates for 9 weeks. In the end, I assessed native diversity, non-native, and also cryptogenic. I was interested in, of course, in taxonomic diversity, but also functional diversity as well. Functional diversity was assessed based on this table, which was developed by my PhD advisor in Germany, Martin Wall, and myself. And these are traits very important for fouling organisms, based on body size, growth form, trophic type, modularity, and motility. Just to give you an example, the famous Bottele Schlosserei, this Ascidian. So as for body size, it's within the large category. As growth form, it's an encrusting organism. Trophic type, it's a suspension feeder, it's a colonial animal, and it lives attached. So it has this set of 5 letters, and this constitutes one functional group. So several species could share the same functional group. This is just an example. Okay, results. This looks chaotic, I know, but the idea is the top figure is control, disturbance 1, disturbance 2, and disturbance 3. This is just to show you that the level of complexity of these plates. So you have a more complex community and diverse community and my control, and in opposition, in contrast, you have on disturbance level 3, with high copper, we have apparently less diversity, okay? But this is just how it looks. We wanted data, so we wanted to see if our treatments were indeed working. And what you see here is water samples. So after 3 weeks and 6 weeks, I took the plates out of the water and put it in buckets with water. And after 2 hours with the same volume, I took water samples. And I found a perfect, actually, gradient of copper. So you see with the white bars are control, and then increasing with the gray scale, we have disturbance 1, 2, and 3. And this means that copper was indeed leaching from the anti-fouling paints. Also with the biota, so we wanted to check if the fouling assemblages were assimilating copper. And this second graph shows that yes. This is an example with the barnacles. They were colonizing all plates. And it shows that the tissue of these barnacles were assimilating copper across treatments. So our treatments were working. And this is what we found in terms of diversity, non-native, native, and actually total. So this is just Panama. The graph on the left is for taxonomic richness, and the graph on the right is for functional richness. The white line is actually total diversity. Green stands for native, and red for non-native. And the levels of disturbance on the x-axis. So we found that in Panama, all types of diversity decreased with copper, and were significant, including with non-native. Virginia, similar pattern, but not for the non-native species, and the reason is we only found 2 invaders. So the signal was very, very low. So finally, main conclusions. Total and native diversity decreased with increasing copper for both taxonomic and functional richness. Non-native diversity decreased with copper exposure at the tropical site, and this contradicts previous investigations. The invasive signal at the temperate site was too low to detect any effect. And this may be because of the invasive species pool in a certain region may play a key role in shaping these patterns. In fact, this was just a pilot study, it's already published, and we expand the study to several regions in North America. I'm still analyzing data, but I can tell you now that this pattern is consistent across regions. Thank you very much.